All right, guys. So I think I'm going to be able to finish up here. We'll see. Let's hope so. So it's only 3 videos. So a little bit more about inhibitors. Okay. We know the difference between competitive are going to block the active site, right? Blocks directly. Okay. We can even say this is direct. Okay. Non-competitive is indirect, indirect, because it will attach to another location. That's called that allosteric location. And it results in the enzyme changing its shape and changing the shape of the active site. So here we have a bunch of examples, and I had to stop myself because some of these were interesting, but they all seem to be like kind of bad things at first, or things that were having a negative effect. And generally speaking, inhibition is not a bad thing. We'll get to that, very important saying that I say down here in a moment. So let's focus on the first one here, okay? And that's sarin gas. That's like nerve gas. Years ago, somebody actually released sarin gas in like a subway in Japan or something. All these people died. It was absolutely terrifying. It was like domestic terrorism or something. It would be absolutely horrible if somebody got a hold of this. Sarin is a nerve gas that actually binds to the active site. So would it be direct or indirect? I'll wait. Right, it's direct. Okay, I hope I didn't startle you. Because the sarin gets right into the active site of this thing called acetylcholinesterase, 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 acetylcholinesterase. Acetylcholinesterase has in its active site, it has serine, not sarin, serine, which is actually an amino acid. And sarin actually attaches to the R group. Remember the amino group, the carboxyl, the H, and then there's the R group. I don't know what the R group looks like off the top of my head, but the R group. Sarin attaches to the R group of this serine that's in this active site of acetylcholinesterase. That's the enzyme. So this is going to be a direct competitive kind of inhibition. Well, what does that do? When it blocks that, right, then it blocks the production of acetylcholine. It blocks the breakdown of acetylcholine. Big mistake there. Okay. You want to break down acetylcholine in your body after it's used. Acetylcholine is produced so that a nerve will skip one nerve and go to the next nerve, or a muscle contraction will go to the next. But you don't want your muscles or your nerves to keep firing, right? So that's why acetylcholine, after a nerve fires or after a muscle contracts, acetylcholine needs to be get out of there, and broken down, or else your nerves would continue to go, go, go. So this is a terrible death that somebody would have, because acetylcholine, since it blocks the active site of that, acetylcholine stays present. And the nerves keep firing and firing, producing even more acetylcholine. A person that goes through this, their muscles literally all contract up on them. They usually die from their heart muscles and their lungs just contracting up on them. But it's also their nerves keep firing. So it's a very painful, horrible, it's usually a quick death. But sarin, nerve gas, this was what was used in a lot of times in Vietnam. And it's one of those biological chemicals that I believe are kind of, not acceptable uses anymore because of like the horror that it like causes. Okay. And the pain, and it's unnerving. No bad joke there. Okay. It's actually nerving because it actually makes the nerves fire more and more and more. Okay. So sarin is one of those things that uses competitive, which is direct. It blocks the active site from this thing being able to break down acetylcholine. And you want to break down acetylcholine when you're done. Now, DDT was a big thing way back in the 70s when they finally banned it. But before that, DDT was actually discovered in the 1800s to be a chemical that would help kill mosquitoes. And it inhibits ATP synthetase actually in mosquitoes. So mosquitoes can't form the ATP synthetase. They don't have any energy. They die. right? That's how DDT works in mosquitoes. But in humans, and here's like the bad thing about DDT. Back like when my mom was a girl, they used to have these trucks go down in the summertime just spraying DDT gas, right? And the kids would follow the trucks. They'd be like dancing in the gas, like dancing in pesticides. Like they just didn't know back then. That's like back in the 40s. And a lot of these kids were exposed to this DDT. And in humans, it causes, like there's three big things that it causes here. It causes sodium ions channels to be delayed. And here's the thing. We're wondering, is it competitive or non-competitive? It's a non-competitive thing. So that's right. This one was competitive up here. This one was non-competitive down here because it's going to hit an allosteric location on these sodium ion channels. Also on the potassium gates, hits an allosteric thing that changes the shape of these sodium ions or potassium gates. And these are the things in nerves, the sodium potassium pump, and in muscles, okay, that actually help your nerves to fire. So it would actually stop. And people would get twitches, like kind of after being in the DDT gas. They'd have like tremors and twitches. And eventually it would go away. But the DDT would break down a little bit, but would also bioenhance too later. We'll get to that. The other thing they found later that DDT actually affected were calcium channels. Calcium channels are usually at the end of a nerve. Okay, that's like how a nerve transmits one signal to the next nerve, and they found it like triggered that too. So what happened is, here's how it actually happened. Oh no, this is the birds coming up. Here's how it actually happened. Once all this stuff, the sodium potassium pumps were not working, the calcium channels weren't working, then the nerves weren't firing right. And they found that it would remain in their systems and not only cause these tremors and almost like Parkinson's like syndromes, but then eventually it built up and it caused cancers and stuff. And pancreatic cancer is a big thing that was affected by DDT. People were affected by DDT exposure. Okay, now in birds, that was like when the big cry came out. This woman wrote this. Silver Springs, I think it was called. Oh, I'm terrible that I'm not remembering this. But they sprayed a lot of DDT up in New York, and they noticed that what was happening is the birds were dying off, like eagles especially. Eagles were going to their nests after they'd lay the eggs, and as the embryos developed inside the eggs, they became too heavy for the shells, and the shells would just literally crack. And so mother eagles were coming home to like scrambled eggs in their nests. I know that's a bad joke. But they'd come home to all these eggs that were cracked. And it was the reason that the eagle population declined, and the bald eagles we're talking about, not golden eagles, okay, but golden eagles too. A lot of birds were affected by this. But the bald eagles were the big thing. They're our national symbol. And when they started to decline in numbers, they saw that it was related to DDT. Now, how this happens, though, is DDT actually is converted to something else called DDE. And that DDE is actually what would inhibit calcium from being distributed into the eggshells. So calcium has to come out of the bloodstream and has to diffuse into the eggs as like the female is releasing her egg to make the egg hard. And when this DDE inhibited that, it was again a non-competitive inhibitor, inhibited this calcium chain, this calcium ATPase, a little different than regular ATPase. It prevented the eggs from becoming very hard. So when the embryos grew to a certain size inside the egg, it just couldn't hold it anymore and the egg would fall apart. Okay, and it's amazing how they find these things out. Okay, so again, it was DDT that got converted to another form called DDE. Don't ask me the long names of these things. It's like but anyway, these chemicals, not only in humans was it causing these nerve problems and cancers, but in birds it was causing their eggs to thin out and break, and populations were declining. So those are two very bad things. So we had to have something good to talk about. And penicillin, okay, I know it's a mold, it's a fungus, but like what's so good about it? It works as an enzyme inhibitor too. It inhibits this enzyme called transpeptidase. What is transpeptidase? Well, it's actually an enzyme that forms these cross linkages in the type of cell wall that bacteria have. They have this substance called peptidoglycan. You might remember that. That's a substance that's in the cell walls of bacteria. And transpeptidase helps peptidoglycan form. But penicillin inhibits that enzyme, so the peptidoglycan can't form, the bacteria doesn't get a cell wall, and the bacteria die. So when you take the antibiotic penicillin, it inhibits this enzyme that the bacteria produces that helps to build its cell wall up and make it real tough. Okay. Instead, its cell wall becomes loosey-goosey like the poor chicken eggs or eagle eggs, excuse me, and it kind of breaks apart. So the bacteria literally kind of like fall apart like the eggs did. So just like DDT weakened the eggs, penicillin weakens the cell walls of bacteria, and that's how it works. And it works as an inhibitor. Okay. Finally, inhibition. And I'm going to give you still one more example. We have to say inhibition. We're talking about all these things that it stops. That might be like good things. I mean, this is a good thing for a bacteria. Yeah, he wants a cell wall. Okay. And these are definitely bad things. Okay. But inhibition is important in controlling metabolic pathways. How? Because when you produce something, we want it to stop once you have enough of it, right? It's kind of like if somebody gives you a pen, right? And you're like, okay, thank you, I got a pen. Oh, no, here's another pen. Oh, no, here's another pen. Here's another pen. Here's another pen. What do you have to do to make the pen stop from coming along? Here's another pen and another pen. You're like, stop. Okay, so sometimes once you have one pen, it's enough to stop anybody from giving you another pen, right? You have a pen, you're writing with a pen, you don't need another pen. So that's where we're going to go into the next thing. And I'm going to give you a peek of this, but I'm probably going to stop this video before I talk about this in detail. And it's called feedback inhibition. How that positive, negative feedback, primarily negative feedback, is going to help stop something. When you're making something, you want it to stop eventually. Sorry, you can't see that at the top there. And I'm going to stop there. It's a good stopping point, and pick that up in my next video.